nucleus, and it's basically tricked. It thinks it's been fertilized. Right? That's the basic idea. And then if you reimplant it, it can launch into a developmental program, and it will play out in exactly the same way as the original parent. Yeah, question?
Okay, so, I mean, the process in itself sounds complicated, but, I've heard a lot of, like, people attempting to have cloning thing on animals, and it failed. Like, they're trying to bring back an animal from extinction or something, and it failed. What's the exact mechanism that causes that to fail?
So, I mean, first of all, bringing back an extinct animal is going to be hard because you don't have the nucleus, and the DNA can be fragmented and so forth, right? So that kind of Jurassic Parky thing is maybe a little bit less likely because those big subcellular structures deteriorate real quickly, okay? But there's other reasons it might fail, and I'll get to that in a moment. But some of the reasons it might fail is because the genomes haven't gone through spermatogenesis and oogenesis. So I'll talk about that in just a moment. But so if we want to look at a kind of a picture of what's happening here, so I know it's annoying when someone does this, but you remember this. In this case, the idea was we took the nucleus from the mother who wanted to be the mother, and we put it in an oocyte from a donor. The donor oocyte provides the mitochondria. The mother's nucleus provides 1/2 of the diploid genome. In the case of cloning, it's pretty similar. You have a recipient egg where you remove the original nucleus from that egg, but instead of putting in the nucleus from the mother's oocyte, what you do is you put in the nucleus from a differentiated diploid cell. So we could have, let's say, another pathway here where we take the nucleus out of a somatic cell and we put it into this cell where the nucleus has been removed. So now it's automatically diploid, and the oocyte thinks, hey, I've been fertilized. The oocyte contains all sorts of reprogramming factors. It tries to strip off everything on these two genomes from the somatic cell, and it tries to reset the DNA in some sort of original totipotent state so that you can develop all the way again through development. And then what you get is programmed by the DNA that you put in there, which came from some parent organism like Dolly or something like that. So this figure really applies pretty closely to this somatic cell nuclear transfer as it does to this 3-parent family situation. Okay, so does it work all the time? No, it doesn't work all the time. The efficiency varies quite a bit. One reason why it might not work is that the 2 genomes that you're starting with in that diploid nucleus have been set in such a way, epigenetically, to drive the identity of that particular cell type, right? Some genes are on, some genes are off, depending on what type of cell you got the nucleus from. But in embryogenesis, what normally happens is you have a genome that's gone through spermatogenesis, a genome that's gone through oogenesis. They've been kind of stripped clean, and things have been re-added correctly in a male or female specific manner. Those genomes combine, and they're ready to go, right? They're totipotent. You can now form all the tissues of the body. What you're asking in cloning is for a differentiated genome that's been locked down in certain ways to kind of be stripped clean and be reset and go through development. And sometimes this works, and sometimes it doesn't. And it highlights the importance of some of these types of processes where you imprint things or you affect the methylation patterns of genomes in male and female specific ways. That doesn't happen in the somatic cell nuclear transfer. And I think what's amazing is that it actually does work, that there's components in the oocyte which say, okay, I see these genomes, but they're not right yet. I need to strip them clean of all these other prior imprints and reset them and see if we can kind of relaunch and get things going developmentally. Okay, so that's cloning. That's a pretty surprising thing that it actually can work, right? Okay. Another example of something related having to do with epigenetics has to do with the ability to reprogram cell identity. And so if you think about development, you start with a 1-cell zygote that's diploid, and it is totipotent. It can form all the possible differentiated cells. It can move down all these lineages to form skin cells and brain cells and everything else. And so the genome that ends up in differentiated cells is exactly the same from cell to cell to cell to cell, but certain genes are locked down and not expressed. Other genes are open and are expressed, and that's going to vary from each different type of differentiated cell, right? And that's an epigenetic phenomenon. The sequence is the same in all those cells, but what you're doing with it in terms of chromatin packaging and so forth varies along each of these developmental lineages. So that's interesting. And what people always thought was that differentiated cells were locked into that particular state. You could never change them. You could never take the genome, even though all the genes are there. You could never take that, and you could never go back again or reroute it. You were kind of locked in that spot. And the exceptions were always things like the starfish, right? Starfish, you chop off an arm and you see it regrow. So it kind of goes back to a stem cell state for that arm and makes it all over again. Or lizards' tails or something like this, right? But you can't cut off a finger and regrow it, right? It doesn't work. And we've always believed that differentiated cells are locked in that state and you can never do anything again. Turns out that's not right. 10 or 15 years ago, 12 years ago, 2012 this won the Nobel Prize, but a number of years ago, this guy Yamanaka discovered that no, you can actually reprogram cells and change their identity, which was a shocking idea. So what you have in this picture is a whole bunch of different types of differentiated cells shown in these various kind of cartoonish color patterns to show you that different sets of genes are expressed in each cell type. And the idea is, can you take any of these cells and revert it back to some earlier embryonic state? Can you strip off all these marks and change it again? And the answer is, it turns out yes. And what he did was he overexpressed 4 transcription factors. These are famous factors: OCT4, SOX2, MYC, and KLF4, OSKM. He expressed these in a differentiated cell and turned them back into an embryonic cell. So that's remarkable. So what are these factors? These factors are normally present in embryonic stem cells. These are the factors that say this cell identity is embryonic. It's open for development into everything, okay? And what he did was ask the question, well, could you take those transcription factors that keep a cell as an embryonic cell, express them in a differentiated cell, and would they cause the differentiated cell to revert back to an embryonic state? And the answer was surprisingly yes, you can do it. The efficiency is low, but you can do it. So this kind of permanent lockdown that we thought differentiated cells were in isn't true. You can undo it, you can release it, and you can move them back again. Yeah, question?
Did he do it in like a living organism, or did he take like a tissue sample and then do it in that tissue?
So initially what he did was he did it with cells in cell culture, and you can look at them and they have different visible characteristics or molecular characteristics you can assess. So we can start with a differentiated type, and then you can see it goes back to an embryonic type. But you can take the embryonic cells that you produce and you can implant them and they'll grow up into an animal as well. So you can do these things in animals also. So it's an alternative mechanism or approach to cloning compared to the previous. So the procedure is shown from the top here. The idea is you have an adult cell that's differentiated, like one of these things, and you add these reprogramming factors, OCT4, SOX2, MYC, KLF4, and they're shown as these kind of these blue things here, but they're proteins or they're genes that are expressed in the cells. The cells now overexpress these embryonic transcription factors, and they turn into something called IPS cells, which stands for induced pluripotent stem cells. And if you think about the words, it makes sense. They're induced, right? You're causing them to change through the addition of these transcription factors. They're pluripotent. That means they're developmentally very primitive, like stem cells, and they can develop along any of these lineages here, right? Stem means, it gives rise to a branch of cells of a particular lineage, and then cells is cells. So IP cells is what you produce, These cells are like embryonic cells. You can implant them, you can grow an animal, or you can cause them to differentiate along these lineages. And people are interested in this because the idea is if a patient has a tissue that's broken or removed or who knows what, is it possible for you to take cells and revert them to an earlier state and regrow the tissue? That's kind of the kind of holy grail of this type of thinking. So anyway, this kind of revolutionized things because it said the genome is expressed with certain patterns in some cells, but you can remold that and cause different expression patterns by changing these epigenetic marks. Nowadays, you don't have to go all the way down. You don't have to take a differentiated cell, go to an embryonic IP cell, and go back up again. People show that you can go directly across. So that's kind of interesting, right? Maybe not everything is allowed, but there's lots of cases where you can convert one cell type directly to another cell type, and you don't have to kind of go down and back up. Okay. Interesting stuff, right? Parthenogenesis. This is weird. Parthenogenesis is a situation where a female who has not been fertilized by a male still gives birth. And it happens in certain organisms, and I have some examples here. One's a shark, okay? And another one is one of these Komodo dragons. And the idea is that somehow the female spontaneously caused an oocyte to think it was fertilized, think it's diploid, and then go ahead and develop into a new organism. And what I've got here is some background that it doesn't happen very much. It's a few species. You have to be careful that the female didn't store spermatocytes from some prior mating, but these examples are pretty well validated. They maybe were animals grown only in captivity, or you could genetically prove that the offspring came only from the DNA of the mother. So you might think about that, right? You could determine through these forensic type techniques that the contribution was only maternal, there was no paternal contribution. So some background. Here's some news report about a captive shark had virgin birth. And then here's a report in either the magazine Science or Nature a number of years ago. And it's kind of interesting to look at this and read this abstract. It says, parthenogenesis, the production of offspring without fertilization by a male, is rare in vertebrate species, which usually reproduce after fusion of male and female gametes. And here they're using some molecular approach, genetic fingerprinting, to identify parthenogenetic offspring produced by two female Komodo dragons. This is one, Flora. Her name's Flora. One of these females subsequently produced additional offspring. This reproductive plasticity indicates that female Komodo dragons may switch between asexual and sexual reproduction depending on availability of a mate and so forth. So interesting types of things. And what you should do in thinking about this is, imagine the question of whether are the offspring identical to the female parent. You should think about this question. It's an interesting question. Are they two clones like we saw with cloning and the IP cell business? Are they clones, or what's going on exactly here? But the idea is the oocyte, which should normally be haploid, probably retains one of those polar bodies in some way and doesn't release them all. And so at some diploid stage of oogenesis, the cell thinks, okay, I'm going to activate at this point and go ahead and make an organism. So something really odd and striking and poorly understood. Okay, what else? You mean, does this involve a somatic cell adding itself in some way to the oocyte? Is that what you're saying? Your guess is as good as mine, but I rather suspect the haploid oocyte retains some polar body type of thing, and that makes a little bit more sense to me. Yeah. Okay, so we have now an argument that I've artificially set up between Mendel and Lamarck, neither of which is here, so I can do this. So Mendelism is what we've talked about so far. Here the idea is particulate factors or genes are contributed by the parents, and the shuffling of these things in the offspring generates offspring with new phenotypes, right? That's the classic genetic model, and that's basically correct. Okay? Lamarck, the idea is that phenotypes that are acquired by an organism during their lifetime tend to get transmitted to the offspring. And so kind of classic or cartoony examples of the idea of Lamarckism is that giraffes get taller and taller necks because they stretch to find leaves that are higher and higher. Okay, so that's one. Another one is blacksmiths by hammering have offspring with stronger arms. I don't know any blacksmiths, but anyway, you see these types of ideas, okay? And the question is, well, is that right or is that wrong? And generally, we have discounted this Lamarckian thing pretty much and gone with Mendel. But I just want to point out that these days with epigenetics, there's some more interest in this kind of Lamarck-like idea. The idea that diet or environment or something might impose controls on DNA sequences through methylation or chromatin packaging in a way that would control their expression and in a way that might actually be heritable. So there are examples of this in simple organisms where treating the organism or growing it in a certain way results in a kind of permanent effect that can be transmitted to the offspring. Okay? There's not a lot of examples, but there are cases like this. And we have mechanisms to explain it, like methylation and chromatin control and so forth. So the answer here, by and large, Mendelism is the basic idea and is correct, but people are interested in a little bit this notion of environmental or context control over genes and the transmissibility of that through these epigenetic mechanisms that we've been talking about. Okay, so answer is sometimes yes, Lamarck seems to be right in this case. Okay, a couple other things. Here's something weird, just for curiosity's sake, I guess. It's called a bilateral gynandromorph, and it's a fly, a Drosophila, that's half male and half female right down this axis here. So that's pretty bizarre. The female side is here, the male side is here. And the idea is the fly started out probably as, let's say, X, big W, big M, and that would cause dominant for red eyes, dominant for normal wings. The other X was recessive, small w, which would be white-eyed, and small m, which would be miniature wings. Evidently, something happened, probably very, very, very early after fertilization, causing in some cell division the failure to transmit both of these correctly. Okay, so maybe one was lost, or we can imagine some scenarios. But ultimately, what you imagine as having to have happened here is that the cell initially was this, but maybe after the first cell division, one of the daughter cells was still this, but the other daughter cell maybe lost one of the X chromosomes. And if they lost this chromosome here, then that side of the body would be male because it would be XO, right? The other side of the body would still be XX. So that kind of a thing might explain this phenomenon here where you've got normal wings and red eyes and a female body type here. And on this half of the body, you have a male body type because it's XO, and it's exhibiting the recessive small w for white eyes and small m for miniature wings. So something pretty interesting, pretty bizarre. Okay, and the last one, I found this a year or so ago. Somebody took pictures of a cardinal in their backyard, and you know, cardinals are supposed to be males having all this bright red coloration, and the females are more tan. And here you have an example of a cardinal that looks half male, half female. This picture here is actually pretty striking. And you say, well, yeah, sure, somebody photoshopped it or something like that. But I think there's a number of examples. If you search for these things, and I've been told by a student before that in birds, this is not so terribly uncommon. So evidently, this is a case where the organism turns out to be half male here and half female here. At least that's what seems. I don't know whether it sings or not. Sings softly to itself. Okay, interesting things. So that's it for these non-Mendelian things. Again, a lot of these topics aren't in the book, but you need to be aware of them because this is what people are excited about these days, and they really extend our thinking away from the stuff we've been talking about so far. Okay, so next thing, population genetics. So we get back into the nitty-gritty of genetics and numbers and so on and so forth. So what we've done so far is we've looked at genes and determined how the chromosomes are going to be given to the offspring. We've looked at inheritance, and we've looked at what genes do, their dominance, recessiveness, how genes or alleles play out in terms of creating phenotypes and so forth. All that stuff is fine and good, it's all right, it's interesting. Now we're going to kind of step back a little bit and look at genes as though they were just entire huge collections of things in the environment, and there weren't, let's say, any bodies associated with them. They're just kind of things out there that we could pick from a population without worrying about the physical context. So population genetics focuses on this idea that we can just consider genes and alleles as numbers in a population without worrying about all the mechanisms involved in transmission. Okay? And we can just pick things, and from the particular collection of possible alleles in a population, we can pick genes out, and we can make one organism or another organism or another organism that's going to have characteristics depending on our chance of picking things from this big pool of genes and alleles. And the other thing we'll look at is the idea that the frequency of alleles in a population may change through time. So if you consider, one snapshot of time, you have certain allele frequencies, and your chance of picking alleles and creating organisms with particular characteristics depends on the frequency of those alleles in the population at that time. But if you go 100 or 1,000 generations later, allele frequencies change. And so then if you pick alleles out of the pool of genes in that later population, you might create slightly different character organisms because your allele frequencies have changed over time. So we're going to look at this idea in population genetics as well. So what can we do with it? So this is a pedigree that we looked at very, very early in the course, and this is an autosomal recessive pedigree. I just want to point out that in this side of the pedigree, this family, we know that some recessive allele, small a, is present in this individual. Okay, so we know the frequency. And let's suppose this individual, they might be homozygous normal, which would cause this person to be big A, small a, right? We know how these things are working. So this is a known here. The problem in these pedigrees is, what do you do with this person? Does she have the recessive allele, or does she not have the recessive allele? And on the one hand, we could say, well, maybe this trait is super, super rare. So it's really, really, really unlikely, and we can discount the possibility that she has it, right? Now, in this particular pedigree, we know she has to have that recessive allele because the offspring are affected, and we're told it's an autosomal recessive trait, okay? If this is something like a blood group, then yeah, we have no idea what she might be because those blood group alleles are present at pretty high frequencies. If you put your hand into the vat and pick out one of those alleles, you really don't know what you're going to get. It's going to be a reasonable frequency. But if it's a severe recessive condition, in all likelihood, she doesn't carry it. Well, with population genetics, we can start to answer this question of what's the likelihood that he, which we know has a recessive allele, what's the likelihood he marries somebody who also carries a recessive allele of that particular type? And that's kind of one of the powerful things that we can think about in these population genetics problems. So just to highlight the fact that this person is a bit of an unknown so far in terms of how we work these, but now we're going to start to ask questions about, can we quantitate the likelihood? Okay, so alleles and allele frequency, we need to do some really, really basic calculations and understand some basic concepts about alleles and allele frequency first. I have a couple cases here, 4 cases. We might have one gene and it comes in only one form, right? So now that particular allele of that gene is present in 100% of the population. That's the only thing that's available in the population. So if you were to imagine, picking out a particular allele of that gene, you'll only get that one sequence. Okay? Now this case is not realistic. We know that gene sequences are long, there's lots of nucleotide changes, but nonetheless, this is kind of way over on one side as at least a possibility. A gene that comes in only one form. Everybody has to have it in that case. Case 2, a gene comes in 2 allelic forms, big A, small a. One possibility is they're at 50% each. So if you say, okay, I want to grab one of these A genes, what am I going to get? Well, you've got 50% chance of picking big A, 50% chance of picking small a. That would be a situation of one gene, 2 alleles, where they come in exactly equal frequency. Well, that's pretty unlikely as well. We could have case 3, where we have one gene, 2 allelic forms, but they're not of the same frequency. That starts to model, let's say, recessive genetic diseases a little better. In this case, I say one allele is present at 90% of the time, the other 10% of the time. Now you think about picking out one of these randomly from the population. In all likelihood, you're going to pick the big A, and only infrequently 10% of the time will you pick the small a. Okay, so that's another intermediate possibility. In case 4, I just wanted to point out that in reality, genes come in lots and lots and lots of different allelic forms. We saw this with the hemoglobin variants and so forth. And so your chance of picking any given allele for that particular gene depends on its frequency overall in the population, right? So we strip away the bodies, we put all these alleles into a big tub, and we just pick things out. What's your chance of getting any particular allelic form? It depends on the allele frequency in the population. Okay, and the point at the bottom is kind of, but wait, it seems like you can only get the alleles that your parents had or have, right? And in a sense, that's true. An individual can only get what their parents had, but the parents too are products of whatever alleles were present in their environment. So you could view them as also having gotten a selection of alleles from the population in some random format as well. So in population genetics, we're really not personalizing it. We're just saying there's no people, there's just genes and alleles floating around, and we're going to collect them based on their frequencies in these populations. So when you think about this, don't think of yourself. Think of just DNAs that you're pulling together from these vats of DNA. Okay, another fairly straightforward point is that the allele frequencies and the genotype frequencies have to add up to 1. And this is a real simple point, but sometimes this provides a check on some mistake in a problem or something that's weird in a problem. The idea is, if you have a gene that comes in a set of alleles, you have to add up all the alleles that's equal to 100% of all the possible alleles in the population, or equal to a value of 1. So for instance, if you have allele 1 of some gene at a frequency of 70% or 0.7, and allele 2 present at a frequency of 25% or 0.25, and that's it, then something's wrong, right? There either has to be another allele to account for the missing 5%, or there's some problem with this, how it's stated in the first place. So once in a while, it's useful in these problems to just assess that everything adds up right. You can't have less than the whole, you can't have more than the whole when you talk about the collection of alleles in a population or the collection of genotypes in a population. Okay, so keep that in mind when you do these problems. It doesn't matter whether we use 44% for frequencies or 0.44. Usually we'll use them as an out of one basis, but the meaning is the same. Okay, so we have a few simple calculations that we should do to determine genotype frequencies and allele frequencies in a population. And the first one is, how do we determine genotype frequencies? We have a population, we have a gene we're interested in, we would like to evaluate how many organisms are of this genotype or that genotype or some other genotype. So the first example is real simple. Let's suppose that we have a gene A that comes in 2 allelic forms, big A and small a, And so we have three possible genotypes: homozygous dominant, heterozygous, and homozygous recessive. And we would like to determine the frequencies of organisms of these three genotypes in some population. Okay, so what do? You got to measure this. It has to be given to you, or you have to measure it. Okay, so you could collect organisms. You could go out and set traps, like this weekend, right? And then you have to separate the organisms into genotype classes, right? So how do you do that? One is, maybe you could just observe them, right? That's the easiest. So if these were the Andalusian chickens, these would be black, gray, and white. So you could just look at them and you could bin them into these categories and you would get some numbers. The alternative would be, well, you could use some protein detection method, or maybe some molecular approach like PCR or sequencing, something like that. But whatever technique you use, you have to be able to measure what the genotype of the organisms are. So now let's suppose we do that, and we find out that 78 out of 100 are homozygous dominant, 20 out of 100 are heterozygotes, and 2 out of 100 are homozygous recessive. So it's just numbers. You have to be given them, or you have to measure them. It's not something that you just create out of nothing. So in this case, the frequency of organisms with genotype big A is 0.78, frequency of heterozygotes is 0.2, and the frequency of homozygous recessive is 0.02, or 2 out of 100. Really simple. Okay, you measure things, or you're given things, and you determine genotype frequencies, and you want to maybe check that all the collection of all the possible genotypes actually adds up to 1. So the frequency of these plus the frequency of these plus the frequency of these ought to be 100%, or a value of 1. So simple check on it. Okay, genotype frequency. What about allele frequencies? Allele frequencies we can do a couple different ways. The first one is kind of clunky, where we add up the number of organisms of each genotype and do some simple calculation and determine allele frequencies in the population. And the other one is a little bit of a variant on that idea. So I think this is from the book, but let's suppose that we've determined the number of organisms of particular genotypes and found 293 of these, 604 of these, and 103 of these. So those would be, out of 1,000, we could calculate the genotype frequencies for each of these categories. How do we get allele frequencies? Well, each of the organisms has 2 possible alleles that we could transmit. So if we take the total number times 2, that's the total number of alleles we're talking about in this population of 1,000 organisms, right? Everybody's diploid, and you can either give this or that. That's the idea of the times 2. So total number of alleles in the population is 1,000 organisms times 2 alleles each. And what we want to do is then compare how many big A's do we have compared to 2,000, how many small A's do we have compared to 2,000. So one way to do it is to kind of add them up. And what we're going to say is that P is the frequency of big A allele, Q is the frequency of the small a allele. And we'll see this type of terminology used again and again in these problems. The frequency P of the big A allele here would be the number of these organisms times 2, since they both have the this or that, big A or big A, right? Plus the number of these organisms. Each of these has 1 copy of big A. So that's pretty easy to think about. The frequency of big A is twice the number of these individuals plus the number of these individuals divided by 2 times the total, which is the total alleles here. And so you get whatever this resolves itself to over 2,000, and we get an allele frequency, P, for the big A allele of 0.595, about 60%. Okay? And you can do the same thing with small a. You take the number of homozygous recessives times 2, since they both have either small a or small a, plus the number of heterozygotes, each of which has 1 small a, add them together, divide by the total number of alleles, which is 2,000, and you get some value of 0.405. So in this, I think, obvious case, we have determined allele frequency of big A at 0.595 and allele frequency of small a, Q, of 0.405. And If those are the only two alleles, they have to add to 100% or add to a value of 1. So this plus this equals 1, or P plus Q equals 1 when you talk about one gene, two allele system. Okay, so great, we get allele frequencies through this approach. Another approach is to kind of do it a little bit differently. We're going to take the frequency of individuals of big A genotype plus half the frequency of individuals of big A, small a. And that may seem complicated or a little bit non-intuitive, but really what we're doing is we're saying we're going to take this frequency plus half of these guys. So we're almost kind of drawing a circle around this half of what we see here. And the next thing we'll do is we could draw a circle around this half. So it's the frequency of these plus half the frequency of these. So frequency of big A is 0.293 from the previous slide. Half the frequency of the heterozygotes is half of 0.604, and you get the same value. The reason you might use one approach or another is just because different problems will give you different information. Right? This problem here is more kind of starting from the initial experiment where somebody collected organisms. This is more often what you would see in a problem where you're just kind of manipulating the numbers around. Okay, and then we know P plus Q equals 1, so rather than do this again, we'll just say in this case that 1 minus whatever we got here has got to be Q, the frequency of small a, which is 0.405, and so voila, we're done. So that's how we do genotype frequencies. You're given numbers of organisms in each category, and you divide by the total, and then we got two approaches to look at allele frequencies. One kind of more clumsy and cumbersome, and then this one was kind of slick, which you can do from genotype frequencies. Some notes. These are important notes. Number of alleles is less than the number of genotypes. Well, I think you already know that, right? If we have two alleles, big A and small a, we can have 3 genotypes. So it's just a note to remind you of that type of a thing. Note 2 is more important. For X-linked genes, females have twice the number of alleles compared to males. So sometimes this can affect a problem, or you have to take this into consideration. And note 3 is, what I've shown you is pretty simple. I think the books, both of them, I think, have illustrations or examples that are a little more complicated involving multiple alleles. So please take a look at those types of things. Okay, so what do we do with that? It turns out there's a famous equation called Hardy-Weinberg equation, which may seem complicated initially, but I think you'll come to realize that it's really nice, really useful, and really you can do a lot with this. The equation is shown here, and what it's trying to do is relate allele frequencies in a population of genes, let's say, with the types of organisms or genotypes that you can produce based on those allele frequencies. And so we've got here an equation where we have the frequency of the big A allele, P, squared, that's one term, that's going to be one category of genotypes, plus P times Q, which is your chance of generating a heterozygote, as another term, and then Q times Q, this is the chance of the small a allele and another small a allele. That's another term, and those are set equal to 1. It turns out each of these terms reflects in this case, the homozygous dominant genotype frequency, the heterozygote genotype frequency, and the homozygous recessive genotype frequency. So this is neat, because if you know P and Q for some 2 allele system, you can use the Hardy-Weinberg equation to make predictions about the number of individuals with this genotype or that genotype or another genotype. The reason it's equal to 1 is that those are the only 3 genotypes that are possible in a 2 allele system. They have to add up to 100%. So the number of big A plus heterozygote plus small a, that's all you've got. So that has to equal 1. Right? So this equation is pretty nice. A population where this equation works, where you can go back and forth between genotypes and allele frequencies or allele frequencies to genotype frequencies, we call that a population that's in Hardy-Weinberg equilibrium. So just maybe a fancy way of saying that. Okay, so how do we think about this? This isn't so intuitive necessarily. Let's make it ultra intuitive. Let's suppose that we have all these possible alleles in a population put in a big vat like this. And in population genetics, this is how we think about it. And like I said, we're just going to reach in and pick out an allele and see what we get. And the chance that we get it depends on P or depends on Q. Okay? And I've divided these into male and female. We can do that or not do that. I've done it here because, in part because their Punnett square approach does it that way in the book. But the idea is we put our hand in and we grab something. What do we get? We might get big A. The chance we get big A is the allele frequency of big A, which is P. Okay? We put our hand in the other vat, we might get big A. Chance is P. So the chance that you get big A is P times P, right? The frequency of the big A allele in these kind of vats. So that makes sense. That's one term in this equation. If we put our hand into this vat and we pull out big A with chance P, but the next time over here we pick out small a with chance Q, that's chance P times chance Q. Okay, that's a heterozygote. But we might pick small a here with chance Q, and we might pick P, or big A here with chance P. That would be QP. That would be another heterozygote. So that's why the heterozygotes have twice PQ. You can pick it two ways. You can pick PQ, or you can pick QP. Okay? And the last one is just like the big A. You might reach in here and pull out allele small a with a chance Q, and you might do it again, and that would be Q times Q, and that gives us the last term over here, and then that's how we get kind of the idea of what we're generating here in genotypes. P squared, big A, 2 times PQ is the heterozygotes, and Q squared is the small a. The book does it in a slightly different way. It basically says that from the male, you might get big A with chance P, small a with chance Q. Likewise for the female, big A with chance P, small a with chance Q. Not thinking about individuals or physical objects, but just vats of alleles, and you combine them together, P squared, Q squared, and 2 categories of PQ. Okay, so I think this is a nice way to think intuitively, or in an illustration, what you're doing in generating an equation like this. Okay, the Hardy-Weinberg equation works really, really well for lots of problems, but we have to make some assumptions. Sometimes those assumptions hold, they're true, or they're good enough. Other times they're not true. So we need to know what assumptions this Hardy-Weinberg equation is making to know when we can apply it and when we cannot apply it, because we can't use it all the time. Okay, so let's look at what assumptions it makes. First of all, I guess mathematically, it makes the assumptions that the population is infinitely big. We don't worry about this very much, okay? It's never infinitely big, but it's big enough. Matings are random. This is an assumption. That is to say, organisms do not preferentially mate with one another on the basis of what their genotype is. Obviously, this isn't completely true, but we're going to make this assumption, this random mating assumption, in order to let us use the Hardy-Weinberg equation to solve problems. So we're going to assume matings are random and not depending on what the genotype or phenotype or anything is. We assume mutations do not alter the frequency of alleles. This is a pretty safe assumption in a short period of time. Mutagenesis happens. It's super important. We'll talk about it a little bit later, but it's not a mechanism to give rise to big sweeping changes in allele frequencies. It's a real kind of accidental, spontaneous event and doesn't really appreciably alter allele frequencies. So that's probably pretty safe. A couple of others that will deal with these exceptions later. We're going to assume there's no migration of organisms into or out of the population that we're dealing with, because that could change allele frequencies if those two populations didn't match perfectly. And there's no selection for these alleles. There's no preferential transmission of big A or small a. Obviously, this isn't always going to be the case, but we're going to make that assumption when we do these simple problems. Okay. If we assume all of this stuff, which is safe enough in many situations, but there's some where you have to be careful. But if we make these assumptions, then in fact it's true that genotype frequencies are p squared, 2pq, and q squared. How much time do I have? Okay, I think I'll do this and probably end. You can map out the relative genotype frequencies of big A, the heterozygotes, and small a on a graph like this, where the axes are showing you the kind of changes in allele frequency. So what you have on this axis up here is the frequency of allele big A with the value of P going from 1, that's all there is, to 0, there's none of this. And in kind of an opposite orientation, you'll have the frequency Q for small a, where you have that's all there is, to that's none of what there is. And what you can see is for any of these kind of pairs of conditions, a value Q and a value P, you can calculate the frequency of all three types of genotypes in the population if it's Hardy-Weinberg. Okay? And so let's say the position here is kind of a unique point where you have half P, half Q. Here you have maximum number of heterozygotes, and you have equal numbers of homozygous dominant and homozygous recessive. So that's kind of a notable position on the graph. For a typical, let's say a typical recessive disorder, some serious recessive genetic disease, in that case, we're way off on one side, right? Normally, then the value of the wild type allele, big A, would be very close to 1. Most of the time, that's what people have. But there's this very, very low frequency of some recessive disease-causing allele that's near 0, but not quite at 0. In those cases, which are typical for these types of diseases, most people are completely normal, genotypically big A. There's a certain carrier frequency of people who are big A, small a, who carry small a but don't exhibit anything. And then there's a very, very low frequency of individuals who are small a's who have particular traits. So way off on one side would be characteristic of typical recessive genetic diseases. Okay, I think I'll stop here. We'll continue discussion of population genetics on Thursday.